going to introduce our lab briefly. Recently, as well-being becomes more popular, demand for functional materials such as carotenoids, terpenoids, and flavonoids is increasing rapidly. The various functional materials have physiological activities in the food, pharmaceutical, and cosmetic industries. However, most of these functional materials are present in small amounts naturally, and their molecular structures are quite complex. So, economical production of these functional materials is difficult with conventional extraction, purification, or chemical synthesis techniques.
We are researching the development of new technologies based on biotechnology for the mass production of these new functional materials. Specifically, we are designing and optimizing biocatalysts and biological process using advanced technologies such as microbio-metabolism engineering, enzymatic engineering, molecular biology, and biochemical engineering. After graduation, we can work at the research center of a pharmaceutical and cosmetic company as well as a famous food company. Next, she will tell you more details.
Hello, I'm Yeonji Kim, a master's candidate at the Food Biotechnology Laboratory. From now on, I will explain about the lab experiments. I'm gonna explain the incubation during the experiment in our lab. We usually use E. coli because E. coli is the most widely studied microbe and an important species in the fields of biotechnology and microbiology. We use DNA cloning to make recombinant E. coli. From the recombinant E. coli, we can efficiently obtain enzyme used for biotransformation. Then first of all, take a stock. This is recombinant E. coli stock we made, and we stored minus 70 Celsius over there in deep freezer, and streaking or spreading it on the solid medium. This is solid medium. Streaking is the method of separating from the various strain to obtain a one colony. We want and spreading is a method used to determine how much bacteria is present in the solid. After this step, it is going to go into the incubator at 37 Celsius overnight. If colony grows on plate like this, colony grows in place, put LB medium and colony in the test tube, like this, test tube, and pre-culture them. The pre-culture required time for microbe to adapt to their new environment, so they need the breeding page before main culture step. You can choose the medium for your purpose. Our lab usually use our LB medium. 37 Celsius is the optimal temperature at which E. coli grows, and RPM is unit of how many turns in a minute, and set to provide the oxygen to E. coli. We call preculture seeding, so this is called seed, and we use this for main culture. All of this should be done in clean bench over there. It is very important to select two suitable growth medium, temperature, and RPM as the preparation for subsequent biotransformation or enzyme reaction. Next part is biotransformation. Thank you.
So far, we are taught you how to grow microbes. I'm going to explain to you how we use these microbes. We can use them for wholesale biocatalysis, or we can isolate enzyme from the cell, then use for biotransformation. And biotransformation is the chemical modification made by an organism on a chemical compound. In our laboratory, we mainly use fatty acids as starting materials. Most renewable oils consist of glycerols and fatty acids. These fatty acids can be cleaved into diverse valuable carboxylic acids such as azelaic acid in this slide. Renewable oils are known to be one of the abundant resources in nature, but in the industry, these oils are low-priced products to get high profit as itself. So it is meaningful to use these materials for substrates and converted them into a high-valued products with enzymes which are provided by microbes. For example, azelaic acids are very stable and widely used as antimicrobial agents like acne treatment. In our laboratory, we designed novel multi-step biotransformation pathway for production of carboxylic acid from renewable oils. There are so many steps to convert renewable oils to products, so from now on, I'm going to explain about the just first step of this process. Oleic acid is major component in vegetable oils. We are already familiar with olive oils, and oleic acid is major component of the olive oils. It is attractive for producing hydroxy fatty acids because oleic acid is really cheap. By enzyme oleic acid hydratase, so-called OHYA, oleic acid gets water, then converted to 10-hydroxystatic acid. Because of high reactivity of 10-HSA, it is very useful. After several steps of reaction, it can produce high-valued materials. Reaction conditions such as temperature, pH, and the cell and substrate concentration were optimized to use recombinant cells containing the oleic hydratase. In addition, because fatty acids are hydrophobic, we also add substance that acts as surfactant. After setting the reaction conditions and mixing the necessary materials, the reaction starts by enzyme. Finally, we can get what we want. Then we analyze the product compounds, and from here, next presenter will continue.
Hi guys, I'm Chaeyoung Kim, and so far you guys learned that we can earn very valuable products from very cheap substrate like oleic acid. But now we don't know exactly what they are, so we are doing the process of analysis. Usually we use a lot of equipment for an analysis, but usually these two kind of equipment is used, and they are kind of chromatography. Do you know chromatography? No? Chromatography means separation of mixture. So there are a lot of things in mixture, like substrate and final products and so on. So by separating the mixture, we can identify what they are. And the left one is gas chromatography. So it uses gas for mobile phase. And how about this? This means high pressure liquid chromatography in some HPLC. This uses liquid for mobile phase. So, and this picture shows very basic principle. And gas chromatography and HPLC also have three parts in common. There is an injection, and the column, and detector. And especially HPLC use pump to have a liquid to move. So now we can get this kind of graph for using the equipment. And we can see 5 peaks over there, like here and here, here and here, here. And 7 hours later, we can find also 5 peaks, but the green one decrease and the red one increase. And the fourth one can be a standard. So we can assume that the green one decrease means substrate, and the red one, the increasing one, means product, final product. So now we can quantify how much products increase and decrease, but still we don't know exactly what they are. So by using a mass analyzer, we can find which peak means which materials. So thank you. so far, the many things that we have been said can be difficult for you guys, but it is very usual thing. So now, from now we will look around the laboratory, and we have also main laboratory over there. But first, see the equipment and see the process of incubating. And now stand up and follow her, please.